Hello, my name is Elyssa Garza, and I'm a graduate student at Texas A&M University. Today, I'll be talking about using comparative genomics to identify serpentine tolerance loci in Caulanthus and amplexicaulis. Now, before I delve into this topic, I would like to mention a little bit about what are serpentine soils. Serpentine soils are harsh soils. They are characterized by low amount of nutrients, low calcium to magnesium ratios. They contain heavy metals such as chromium, nickel, cadmium, cobalt, and they retain low amounts of water. If you look in the image on the right, you'll notice a serpentine outcrop. This outcrop has very little vegetation, and this is evidence of the stresses that this type of soil provides and the adaptive capabilities needed to survive a serpentine environment such as that. Now, what we want to emphasize here is that serpentine soils provide a lot of stresses, and we know that many of these stresses haven't been thoroughly investigated. The plant that I use to understand serpentine tolerance is Caulanthus amplexicaulis, which is a great lab organism. It is a close relative of Arabidopsis thaliana, our model organism. It has a short generation time. It is infertile and self-fertile. We can transform it by floral dipping and tissue culture methods. And it also has a small diploid genome with a haploid chromosome number of 14 and a genome size of 372 megabases. There are two varieties of Caulanthus amplexicaulis that I'm looking at. We have variety amplexicaulis, or CA, which is mostly a granite living individual, and its sister variety Barbarae is actually a serpentine endemic. Both plants are located in California, and we can see that CA has a much wider distribution in comparison to CAB, our serpentine endemic. Now, we have experimented with both of these plants. We have grown CAB and CA in different types of media, in control, nutrient balance media, in nickel media, in serpentine, in granite, and we have seen that in our harsher media that CAB tends to grow just fine, but CA, on the other hand, tends to grow more stunted and it grows as in the images above. When we look at the two genomes, we can easily see that CAB must have some kind of underlying mechanism in order to survive serpentine or harsh in media. However, CA does not have the same mechanism. So together, these impose the question, what genetic mechanisms allow one plant to survive serpentine soils and not the other? The way I choose to address this question is by looking at these more specific questions. Can we identify the loci contributing to serpentine tolerance? And if we break down serpentine soils into the components that make it stressful to a plant, can we identify the QTL involved with that specific element, specifically nickel and low calcium? Here is a simple workflow of the methods I've used in my experiments. I've assembled and annotated the CAB and CAA genomes alongside resequencing 169 recombinant inbred lines. I've identified markers in the parents and real genomes. Using the marker and assemblies, I've created genetic and physical maps. I've also phenotyped CAB, CA, and the RILs in different stressed environments. Together, the phenotypic and genotypic information were used to identify QTL involved in specific stressed media. And then going forward, I'm also using a multiple evidence approach to identify candidate genes. First, we're going to take a small look at our assembly statistics. We can see that CAB and CA both have 751 scaffolds. The shortest sequence length at 50% of the genome, or N50, is about 22.5 megabases, and the longest scaffold is about 27.8 megabases, showing that much of the genome has been collapsed and is starting to reach chromosome level. For our genome coverage, we can see most of the genome has been covered according to its predicted size of 372 megabases, so 89% of the genome has been covered. I would like to make note that CAB did have a lot more information going in than CAA. It had 2 by 250 paired ends and multiple types of mate pairs, whereas CAA only had 2 by 100 paired ends. I did have to reference map CAA to CAB to create a better assembly. I would also like to make note that I will be talking about CAB and CAA at the chromosome level, and we remember that CAB and CAA have 14 chromosomes. In the assembly for the chromosomes, this is going to be covering about 80% of the genome. If we're looking at comparing CAB and CA, I've compared them using a synthetic map created from SynMap in Koji. We can see that CAB and CA are very similar by looking at the diagonal line between the two plant genomes. If you look at the circled squares, those are actual chromosome duplications. If we look at a histogram of the log 10 KS values, we know that KS is important because it's often used to determine the relative ages of synthetic gene pairs. We can use the distributions of these values from the gene pairs to identify unique age classes. So if we're looking at our histogram, we will be first ignoring the primary synteny and noise and focusing more on the other peaks. We can see there was an older alpha duplication event that we predict to be about 50 million years ago, and the more recent event is the mesopolyploid event. If we look at the phylogeny below, it is what we predicted to have occurred. About 15 million years ago, there was an ancestral plant that had 7 chromosomes. Then about 10 million years ago, which would be the mesopolyploid event, that genome duplicated so that CAB and CA now had 14 chromosomes. And then 3 million years ago is when CAB and CA started splitting so that CAB has a serpentine tolerance, but we don't see that in CAA. Further evidence of this whole genome duplication event can be seen by comparing the genes in all of the chromosomes. We can see that many of the chromosomes have large amount of regions that hit to other chromosomes. It's almost 1-to-1, whereas 7 chromosomes hit to another 7, for example, 1-to-10, 2-to-3, and 4-to-8. Now we're looking at how the chromosomes are rearranged. We can take a look by using the block system from Strands et al. 2006. Using his color scheme and blocks, we can see how the chromosomes are very similar to each other and also how they have changed over time by sections of them moving about in between, within chromosomes and on the edges. The incorporation of recombinant inbred lines to the experiment was very important. They were created by crossing 2 highly inbred parents, a CAA parent with a CAB parent to create an F1. The F1 was crossed with another F1 to produce a large set of F2s, which were then maintained through the idea of single seed descent to reach inbred generations from F8 through F12. The idea behind this is that as we are self-pollinating these plants and maintaining the lines, some plants will have a lot of their genes coming mostly from CAB or CAA, and vice versa. And those plants that have more CAA genes will probably be less tolerant to harsh environments compared to those recombinant inbred lines that have more CAB-like genes and should show some type of tolerance. So what I've done is I've, again, resequenced these lines and identified markers. These markers helped us further assemble our scaffolds into linkage groups and also create a refined genetic map. Using these same rails, I've also created a set of phenotypic data based on biomass, chlorophyll, and viability. The plants were grown on low calcium, nickel media, and natural soils, specifically serpentine and granite. Together, the genotypic and phenotypic information were input into QGene to identify QTL. They were determined by permutation testing and a per chromosome significant lot threshold. I was able to use the markers from MSG to identify the physical positions underneath the peaks. Using that information, I was able to build a chromosome map detailing where these QTL are found for their specific stressed media. We can see that 6 of the 14 chromosomes have hits. 4 nickel QTL were found on chromosomes 2, 6, 8, and 12, labeled in blue. Labeled in green is the single low calcium QTL on chromosome 2, and then there are 2 QTL for serpentine on chromosomes 1 and 3. I did find it interesting that the nickel and low calcium QTL did not overlap with the serpentine, And I believe this is because we know that serpentine involves multiple stresses, and it would be difficult to pinpoint all these stresses under one area. In addition, it was difficult growing the rills in natural soils, and this could also complicate the data. Now we can see that many genes fall within these loci. And so what I decided to do is an integrative approach, putting together multiple types of information to isolate candidate genes. I will be using Go annotations, Arabidopsis thaliana descriptions to offer insight into possible functions of these genes by known functions from other plant genes, looking at expression data to support gene expression in the different conditions, and looking at DNDS values to identify positive selection pressures for adaptation, because if things are growing in stressed environments, they're going to have genes that are adapting to help live in these environments. So we're going to be looking for pressures or selective pressure to change. And then finally, I'll also be looking at cis-acting values to confirm that the loci, which are supposedly underlying the tolerance, are actually those loci and not something that is being affected by a trans-acting element or transcription factor. All of these data sets have been integrated together to identify candidate genes, which I'm actually looking at now. So in conclusion, we know CAB and CA have very similar genomes, but for some reason, CAB has some kind of mechanism that allows it to survive serpentine tolerance, and CA does not have this same mechanism. I was able to incorporate genomic data from parents and rills in order to create genetic and physical maps, and using these maps alongside phenotypic data from stress media has given us insight into identifying the loci that are helping with tolerance to specific harsh media. In the future, I will continue to explore the genes involved in serpentine tolerance, and then furthermore validate them via heterologous expression and gene replacement strategies. I would like to acknowledge and thank my supporters, my PI and my lab, also our collaborators and our financial supporters, who of course we can't do any of this without. Thank you for coming, and I would like to open the room for questions.